Major depressive disorder is a psychiatric condition characterized by low mood, feelings of guilt, and suicidal ideation. There are many hypotheses to explain the biological causes of this mood disorder. The most prominent of these is the monoamine hypothesis, which states that a class of neurotransmitter in the brain called the monoamines are lower than they should be. One of these is called serotonin. Commercial antidepressants target the serotonin system to achieve a therapeutic response, but there are several problems with such drugs. It can take up to 5 to 6 weeks for these drugs to have a therapeutic effect. There is also a high rate of relapse among patients taking antidepressants. In 1/3 of depressed patients, these drugs will have no effect. And antidepressants change the brain's neurochemistry, which can have negative consequences. But is there more to this depression and serotonin story? Serotonin is derived from the amino acid tryptophan, which we obtain from the diet. About 5% of the tryptophan we ingest goes towards serotonin synthesis, while the rest is metabolized along a pathway in the liver called the kynurenine pathway for energy metabolism. The enzyme responsible for this is called tryptophan 2,3-dioxygenase, or TDO, which converts tryptophan into a molecule called kynurenine. Under conditions of psychological stress, TDO can become overactive and cause kynurenine to spill out into the systemic circulation. Kynurenine is capable of crossing into the brain, where it can be toxic. Excessive activation of this pathway is twofold. It can cause neurotoxicity and a potential reduction in brain serotonin. In major depression, reduced plasma levels of tryptophan and high levels of toxic kynurenine metabolites in the brains of depressed patients have been reported. My name is Eoin, and I work in the Psychoneuropharmacology Research Group at the Institute of Neuroscience at Trinity College Dublin. The aim of my research project is to establish an animal model of depression that features an overactivation of the kynurenine pathway in response to stress. I am interested in determining how stress-induced kynurenine production from the liver can cause depressive behavior. Following on from this, I aim to determine if inhibitors of the liver TDO enzyme have antidepressant properties. Inhibiting this enzyme has the potential of increasing circulating tryptophan levels and, as a result, brain serotonin concentrations. I use a technique called quantitative PCR, which can measure changes in TDO gene expression in response to stimuli such as psychological stress. Another technique I use is called HPLC, which can measure the levels of serotonin, tryptophan, and kynurenine in the brain. This technique will allow me to determine if inhibitors of TDO can increase brain serotonin concentrations. Drugs which produce an antidepressant effect without targeting the brain itself may provide a more effective and safer way of treating major depressive disorder.